All right. So module two, the biosystems maintenance. We're looking at a lovely.
You should, sir.
Okay, so let's ignore that comment. All right, lovely. So I'll figure it out. But we're looking at the motor neuron, right, in the spinal cord, right, of the ox, right, a light micrograph there. All we're supposed to do is reproduce the diagram, right, an annotated diagram of a motor neuron. Also supposed to show is a diagram of what is here. That's crazy. Bring on a ruler. The ruler is important because how we're going to measure it and get the magnification. How do we find magnification? M is equal to M is equal to
So is it size of giant? Or is
image
it
over
other way around?
actual. Image over actual. I over A. Over A. Mm-mm. So I got my over 80% on my physics exam. I over A. That's always the answer. Alright. Okay, so that's it. So what we're supposed to basically do is reproduce a drawing. I can help you. We know that, right? We're gonna have dendrite. Well, let me just draw a little something, right? I don't want anybody attacking my little diagram. It is my drawing, not yours. Therefore, I don't care. All right. So this is my little drawing of my little soma, and I have my little axon running down. These are my dendrites, right? And this is my nucleus. All the stuff happens there. All the good stuff happens there. And I have my little axon coming down here with my little axon terminals, right? Do not bully my axon has my little myelin sheath channels and here, right? Lovely. I'll move on with our life. It's a rather interesting neuron. Um, but yeah, go ahead.
I can go back up to the drawing, the picture, please.
No, that's not the drawing the picture though, but yeah. So we're supposed to just draw
Sir,
this. Mm-hmm.
how are we? How, what is that? Like, we me just only I see like the cell body. Me not see no.
See
Me
the
not like
axon here?
literally not see no.
That's the
Where?
axon. You see it now?
Oh, oh.
Right, and then we have our soma and our branching out for the dendrite. And then we see a nucleus right here. You can do a little thing, right? It shouldn't be a lot. And then the magnification would be a part of your marks though. Right? So your magnification is a part of the 5 marks. So that should be fine. Say two functions of synapses. What are the two functions that we note? Two main functions. I did go over this. So what are the two main functions?
Release neurotransmitters.
Releasing neurotransmitters and why? So the release of neurotransmitters. That's I guess that's one thing. I guess.
Also it allows for the transmission of nerve impulses between neurons, and it allows for one-way transmission. So I don't, well, based on what we learn, it can go both ways. So I think it allows a transmission one direction, one way.
Mm-hmm. All right, that's fine.
Summation processor.
Mm-hmm. We also have the modulation of the strength. You know that the strength of the impulse actually increases going across a synapse. Do you know that? Mm-hmm. It does. As the electrical impulse goes through the axon, it actually gets weaker and weaker and weaker. So when it goes through the synapse. The synapse allows the we know the process that occurs in the synapse. I explained it thoroughly yesterday. Right. When it goes to the prosynaptic synapse now, right, that rush of sodium ions in that actually revitalizes the signal and allows action potential to continue. Right? That's a part of it. Well, let's look at this. Outline the sequences of events that occurs when an action potential reaches a synapse. Oh wow, the thing that I told you that I described yesterday. Okay, so let's do this sequentially. All right, lovely. What's the first thing? Go ahead.
So I was asking, could you or the person who said summation, could you just explain it? Because I never like I don't remember that.
Okay, go ahead. The person who said summation.
Um, Basically, summation means that you can make multiple decisions at once. So, like if you're driving, you have to accelerate, and plus you have to be looking through this window or that. When do you call it?
Mirror.
Yeah, mirror or that mirror. Plus you have to be turning the wheel, hitting the brakes. The what do you call the stick thingy maloo, all of that. So it allows you
Stick
to
shift.
do multiple things at once.
All right. So summation is generally postsynaptic integration, right? So the postsynaptic neuron, right? It's usually the soma that does that still, right? Then, but at this usually when the signals are taken, right, then a decision is made or integration occurs, right? So remember at the end of the process that I said that there's going to be an excitatory response or inhibitory response, right? And based on the amount of ions that we're letting, whether or not they're cations or anions, will determine whether or not action potential will actually breach the negative 55 millivolts. That's a part of like the summation. It's like a decision making. Like, what do we do? Like, do we do this? Do we do that? Like, what does the cell do? Do we create another action potential? Something like that. So that is what she's talking about. Decision making there, the synaptic summation. Right? So now looking at what occurs here, the action potential. When the action potential reaches the axon terminal, go ahead. Oh, go ahead. I thought I saw your hand up. My bad.
Sorry, what's for this question?
Okay, go ahead.
Sorry, so as you were saying, when the action potential reaches the axon terminal, it's going to open up channels that allow sodium and oh, sorry.
No, you're right. You're right. No, but I like specificity. Specificity is soothing to me. Go ahead. Specify which channel will open up.
Sorry, sodium
Well,
and
not exactly. We know. Okay, I know most definitely that you know that those channels are involved.
Talk, sir.
The voltage gated pathways. That's what I'm talking about, right? Most definitely.
oh
Yeah, but
well
like we're not talking about sodium or potassium. We're talking about activating the voltage gated
okay.
calcium. I heard sodium, right? It's sodium I heard, right?
Yes, sir. You also heard sodium.
Okay, so the calcium gates, right? And what do the calcium do now?
Sir, it's only calcium.
Yeah, generally no. We're looking at calcium particularly because the calcium does something. The calcium is the most important, therefore I emphasize calcium. What do they do? What do they assist in?
The movement of vesicles towards the cliff.
The movement of the vesicles, movement to and attachment of the vesicles. What do the vesicles contain?
Acetylcholine.
Acetylcholine. What is acetylcholine?
It is well, it is the neurotransmitter.
Mm-hmm.
So it simulates the movement of the vessel containing acetylcholine towards the cleft, in which it attaches to the cleft and then release the contents via exocytosis.
Via exocytosis. Lovely. We're gonna have a little journey now across the cliff, right? Have a little
Okay.
journey. Go ahead, go ahead.
Continue, sir.
Oh, you can do it. You can talk about that.
Okay, sir. So now we have rapid diffusion of the acetylcholine across the cleft, in which receptors on the postsynaptic membrane,
What type of
it,
receptors? I love specifics.
ligand gated. That, sir.
Yes.
Sir,
So, mm-hmm.
the
Ligand.
acetylcholine bind with the ligand gated channel receptors and activate them to alter their shape to allow basically sodium ions to move into the postsynaptic cytoplasm. And from there, we have depolarization of the postsynaptic membrane occurring, and that sets off another potential, which continues the basically the transmission of the signal. Now, after that, oh, it just says outline the sequence of events that occurs when action potential reaches a synapse. Sir, continue or just done this? So?
No, that's fine. Yeah, leave it here.
Okay.
That's fine. Right? Because what you're gonna do now is explain the interaction potential thing throughout the postsynaptic neuron. But yeah, so it's there. So outline those. So you're supposed to outline them 6 marks. You could divide this into 6 different steps. How many steps I had? 5? 4? Technically this is a step. Let me break it into more. Technically that is a step. Right. You could speak about the proliferation of the next action potential. Alright, that would be 6 steps. Okay. Um plant possesses transport proteins within cell membranes that move two molecules at the same. You see, this is the reason why the syllabus tells us to just talk about ion movement. This is the reason why I went into symport and antiport and those type of things, because these are the co-transport proteins that we're talking about. One molecule is transported along or down the concentration gradient, which releases energy that is used to transport the other molecule against the concentration gradient. Okay? Differentiate between the term source and sink in flow and transport. What is the source? Guys, what's the source?
They're the source of sugar production in the case of plants.
Alright, and what's the sink? Okay, not hearing anybody say anything. So non-photosynthetic tissue, right? So the thing place that photosynthesis doesn't occur, and the ones that respire and need energy. Okay. Explain how the sieve elements and of the phloem are loaded with solute against the concentration gradient. How does that work? 3 marks. You can draw a little diagram here. You can show your sieve element. And you can show your little plasmodesmata with a companion cell. Right? And you can show your companion cell beside the mesophyll, which is the source. Go ahead. This is your CP cell, your companion cell. And this is your element. Go ahead.
And you're already explaining it, sir.
No, I want you to explain what's happening. I'm just drawing a little diagram. Because you can draw a diagram in the paper, But go ahead.
Okay, sir. So you will have diffusion of the, well, in this case, the assimilate into the companion cell. But after a certain time, you know that the concentration in the companion cell will become higher than that in the source, and with facilitated diffusion, it moves down the concentration gradient. So now the cell will have to switch to active transport in whereby it moves
Again.
ions against the concentration gradient. So we'll have ions moving from in the source, which will now have a lower concentration of the assimilate, into the companion cell with a higher, yes,
You're
a
saying
higher.
ions. Please go back to speaking about sugar or sucrose. Just telling you that you said ions, alright? But yeah,
Sorry,
you're talking
should
about
have been-
that. Mm-hmm.
yeah. And then the companion cell will then have those assimilates moving into the sieve element via the plasma.
Desmata.
Right.
Lovely. Alright, so that's all I need to do explain how it works. That's lovely, 3 marks. And then look at that the diagram that's already prepared. Alright. So we're supposed to discuss the mass flow theory and the mechanism of sugar transport in the phloem. And then your response must include processes labeled from 1 to 7 and the movement of substances as indicated by the arrows. So at 1 we have the source, the mesophyll cell. So we're going to have active transport, right? So we're looking at active transport into the phloem, right? So 1, we have active transport, right? And because we increase the concentration of sucrose here, right, that will actually decrease the water potential in the phloem here, right? And it will increase turgor pressure, right? So the water now would come from the xylem now, right, towards the phloem because of the increase in solute concentration, and that will increase the entire turgor pressure. Now, since here is a high pressure area, right, it will move from high pressure to lower pressure, so I have the movement down here. So the dark arrows are movement of the sucrose or sugar molecules, and the white arrows are the movement of water and minerals, right? So we're going to now move to a less dense area. Right, so the sucrose is going to be moving. I'm putting a lot of dots there so I can see the sucrose molecules moving, right? And then now here, right, we're going to have the uptake, right, the active transport generally again. All right, lately it's going to be active transport. We're going to have the uptake here. Okay, so you're going to have uptake here, right, into the source, right, which in this case is the root cells, right? And because now we're decreasing the concentration of sucrose in this area, right, it will slowly start to increase the water potential and actually allow for water to move back here into the xylem. So I explained the phloem part, right? So so what's happening here now is that sucrose will leave the phloem going into the sink, right? And that will increase the water potential here. But remember, the ions now, the dotted arrows are ions. The ions and minerals moving from the soil will decrease the water potential in the xylem, and it will cause the water to move from the phloem back into the xylem and then move up, right? And that's down the gradient, right? So as the water move from a higher water potential, right, to a lower water potential there, right? Well, higher to a lower water potential, right? Or in this case, we're moving from this higher solute concentration to a lower solute concentration, right? And ions will continue to move up. So that's generally what the diagram is asking for. What you need to do is describe mass flow hypothesis or the mass flow theory, right? Using that. Okay. So you follow your arrows, right? You speak about the change in pressure and you speak about the amount of solute, right? The mass, right? And stuff like that. And you should be able to be well on your way to explaining this theory, right? And this is going to be, hold on, 12, all of 12 marks. 12 marks just for mass flow hypothesis. This is 2021 paper, by the way. Yep. And I think that's it for module 2.